In the human genome, there are approximately 3 billion base pairs of DNA. DNA is essentially the coding for life, and it tells you your body what to do. It tells you what color eyes you have, what color hair you have, how tall you'll be, and many other traits. But DNA isn't always so good. It can also tell your body to develop things like heart disease, cancer, and several other deadly diseases. A possible solution to these faulty DNA is a process called gene editing. My name is Everett Fletcher, and today I'm going to be telling you about what gene editing is, as well as the pros and cons of this process. To start with, let's talk about what gene editing is. Gene editing is a process that allows for the modification of your genome by adding or removing segments of DNA. There are three main methods of editing. These are ZFNs. This was the first to come out. They were the least precise and the hardest to use. The second were TALENs. They were more precise, still pretty difficult to use. And then the next, which is still the most commonly used, and it's the only one used, is CRISPR. CRISPR is by far the most efficient and ergonomic method. It is the cheapest and it's the easiest to use. CRISPR is a programmable protein that can search through all your DNA and find any specific segment. So what it does is it's like a molecular pair of scissors. It cuts that DNA, and then allows for a new gene to be inserted. CRISPR was discovered by Jennifer Doudna and Emmanuel Charpentier, and they're still fighting for a patent for this process right now. Now let's talk about the pros of gene editing. So there's two main applications to gene editing. These are agricultural and medicinal. First, let's talk about agricultural. So currently with gene editing, they can modify the crop or the animal to have any desired characteristic. Some of the main characteristics they desire are things like for them to grow faster and for them to grow bigger. And another thing is for crops, they have them grow with a natural insecticide. So what this is the insect will come and it will eat part of the plant, and this will cause the insect to die. Without this insecticide, it would probably just eat the whole plant and cause the plant to die. This really helps the farmer protect his plants and get them to the market. Another thing it protects against is environmental factors like weather, like hot weather and cold weather. Now let's talk about the medicinal application. So for example, in 1990, a girl named Ashani was diagnosed with a disease called SCID. This was an immune disease that caused her overall immune system to be really weak, and she was susceptible to almost everything. She had to live her life in a bubble up until this gene therapy process came out. So what they did was they modified some of her blood cells to put a new gene in that would fight this disease, and it allowed her to overcome her disease. This wasn't a one-time fix though, and she had to go for routine editings. Now let's talk about the cons of gene editing. One big fear of gene editing is the idea of designer babies. So what designer babies are babies that are pretty much perfect. They're edited to be blonde hair, blue-eyed, tall, intelligent, athletic, all of the desired characteristics you could think of. This is feared that only the wealthy would have this technology, and then put the poor at an even further disadvantage. Gene editing is also very expensive. Even though CRISPR is better and faster than all those other ones, it is still a pretty hard process to use, and it's very expensive. Another thing is the possibility of unexpected mutations. So what this is they make a cut at one spot, and then something else down the line could change. This could introduce a completely new disease to the patient and cause for a really bad time. Before I conclude, I would like to give my sources. My sources are TechCrunch.com, Bionews.org, Raconteur.net, TheGuardian.com, NationalGeographic.com, YourGenome.org, and FAO.org. So, in conclusion, gene editing is a process that allows for the modification of DNA by inserting or removing genes. It has several pros and cons, and I'm really excited to see what path it takes in the future. Thank you for listening, and have a great day.